Change topics just a little bit, Lily. I'm curious, what led you to start researching diets and especially the ketogenic diet?
So I actually think in many cases people come to this direction based on personal issues and personal reasons for being interested. In my case, when I turned 40, I came down with Graves' disease. Actually, I came down is really not the right term because again, like most disease processes, it was probably a very long-term degenerative process that came to a head when I was about 40. And Graves' disease is a dysregulatory disease. It's an autoimmune disease that increases metabolism to an unhealthy degree, gives you very high heart rate, you tremble, etc. And the ways that originally I was treated, so the choices I was given were, either we can irradiate your thyroid, which I wasn't in favor of because the thyroid is a very important regulatory organ. Prefer not to get rid of organs whenever possible. The second possibility was medication, and I unfortunately had bad allergic responses to two of the frontline medications. So I started thinking, what other approaches could I take in trying to manage this disease? And one thing that was a little frustrating for me is that Graves' disease is an autoimmune disease, and yet the ways of thinking about how to treat the disease were very focused on the thyroid. And to me, this seemed a little bit wrongheaded because, again, if it's an autoimmune disease, it seemed like we should be focusing on calming the immune system down. So around this time, I started doing a literature review and trying to understand how autoimmune disease was treated. And I came across a whole series of kind of anecdotal reports that MS, multiple sclerosis, it had been found that a significant number of people who were reporting on this had found some relief from their MS symptoms after going on a ketogenic diet. It did occur to me that maybe I was having some sort of allergic response to something that I was eating, and that maybe this was triggering my immune system to go haywire. So I went through a period of about a year where I was doing blood tests about every 2 weeks or so. And I went through essentially an elimination diet. And what I was looking for was I was looking at my antibodies. Again, this is not typically the way an endocrinologist would think about Graves' disease or what the variable that they would be monitoring. And what I found is that certain foods seem to be amplifying my antibody levels and other foods seem to be decreasing my antibody levels. And by carefully sort of adding and subtracting over time, I wound up following a diet that also, I think not by coincidence, had been successful for these MS patients, which is also, it's an autoimmune disease. So maybe in general, it sort of calms the immune system down. And today, of course, we know that ketones are potent anti-inflammatories. So I wound up already realizing that there was this interesting interaction between different systems, the immune system and other systems. And so initially, my interest in the ketogenic diet primarily had to do with its modulation of autoimmune disease in the immune system. And also, in the context of the brain. And what happened was that over time, I started asking kind of more fundamentally, what is this doing to the brain? One of the frustrating aspects of dietary research is that superficially it seems very accessible to people. And as a result, you get the media picking up stories, eat blueberries, you won't get Alzheimer's if you eat blueberries, or do this or do that. And what I wanted to do was to treat this in a very scientifically rigorous way, which meant really kind of going back to the fundamentals and asking at the most basic level, what are different diets actually doing to the brain? And how do we measure this in a way that is not influenced by people's imperfect recollection about what it is they've been eating? I don't like, food diaries, especially the sorts of research that ask people what they've been eating for the last 30 years, which I can't imagine could be very reliable. And so mechanistically, I really wanted to see, okay, if we change one variable at a time, how is this affecting the brain? And so that led to a whole series of experiments. First of all, we wanted to see if, in fact, there was a dietary effect. So there was no need to get into all of the complex mechanistic studies if, in fact, there was no dietary effect. If it was just a question of confirmation bias, that people just became enamored of some particular fad and then convinced themselves that it was having some salutary effect on their brains, then there was no need to go further. But in fact, the first set of studies looking at fMRI, looking at the stability of brain networks in response to different diets, showed very profound changes between a regular sort of standard American diet and how people responded even after just 1 week on a ketogenic diet. After we had established that there in fact was a strong effect, then we wanted to start stripping away the variables one by one and to start identifying exactly what aspects were responsible for that effect. And that's led to a series of other experiments.
Two key factors linked to age-based cognitive impairment are insulin resistance and glucose hypometabolism. Can you give listeners an overview of how insulin resistance and glucose hypometabolism affect the brain? You sort of alluded to it, but if you could flesh that out a little bit.
One of the features that you see in individuals as they get older, there are a few kind of hallmark clinical features that you see with imaging. One of them that is, at this point, I think, incontrovertible is this hypometabolism that occurs as people age in the brain. And what I mean by hypometabolism is that the brain seems to lose the ability gradually to effectively metabolize glucose. And initially, this was associated with full-blown dementia, but then when people started looking at sort of mild cognitive impairment, they see, that the hypometabolism is something that starts early or earlier in the disease process. And I wanted to understand if, in fact, this was maybe a slow, gradual process that, just as other control circuits, other types of regulatory processes over time start to break down, could it be that what was happening was that this hypometabolism was the consequence of insulin resistance, encroaching insulin resistance, occurring slowly over time? And one of the interesting things about the brain as compared to other organs is that the brain is such a huge, greedy consumer of fuel that it doesn't take much of a deprivation to make the brain respond in a very wonky way. And so I started thinking that maybe the brain was a little bit of a canary in the coal mine, that we all, let's say, tend to become insulin resistant as we become older, but that process might actually be occurring much earlier in the brain because it had such a small buffer in terms of its energy reserves. So from that perspective, initially I was concerned about whether we might be able to measure hypometabolism as insulin resistance in neurons and in the whole brain. But eventually I saw that actually, even in individuals who weren't insulin resistant, and this was very much a surprise, feeding them ketones actually improved cognitive performance and the functioning of the brain, even in the absence of what seemed to be a deficit state. So it's not just insulin resistance. There seems to be something specific to ketones that makes the brain operate more efficiently.
Last year, you had a really interesting paper. I enjoyed reading it in PNAS. It went under the title, Diet Modulates Brain Network Stability, a Biomarker for Brain Aging in Young Adults. The title alone is provocative, and this paper attracted a good bit of attention. In this study, you tested whether dietary changes are capable of modulating sustained functional communication between brain regions by changing their predominant dietary fuel from glucose to ketones, as you just discussed. First of all, can you tell us how this study was conducted, and what were the experiments that you conducted to arrive at the results?
We started first by establishing that diets had an impact on the brain and trying to understand what that impact, if any, what it looked like. Again, I think the problem when you get started with attacking a question such as this is you have to know what it is you're looking for. Because if you don't know what you're looking for, then you can't say if it's changed by one condition versus another. So we started off by putting people, we tested them under normal conditions, people who followed a standard American diet. We tested people under fasting conditions, just an overnight fast, and then we tested them in response to 1 week of a ketogenic diet. And what we found is that the networks in clinical neuroscience and in neuroimaging refer to communication between brain regions, which in practice are measured by correlations between time series in different parts of the brain. And the assumption is if the brain response in one part of the brain has a particular signature and you see the same response in another part of the brain, that somehow they're communicating with one another because they're showing the same behavior. So what we found is that the communication between those different brain regions became more stable, meaning it was more sustained. So those connections, once formed, remain formed for longer periods of time. under the ketogenic condition, as well as during fasting, as compared to the standard American diet. Well, you might ask, okay, so networks are more stable. Is that a good thing? Is that a bad thing? We can't say a priori. So then you want to go and look at what happens to people as they age. And so we looked at some very large data sets comprising nearly 1,000 individuals. We looked at their functional MRI scans. And what we found is that networks start to destabilize actually in the late 40s. It's a sigmoid response, so there's almost no response up until the late 40s. And then starting in the late 40s, they start to destabilize. And it's a kind of a snowball effect that peaks around the mid 60s and then restabilizes around the early 70s. So starting from the assumption that what happens to you cognitively as you get older is not a good thing, particularly as we get to the 60s, 70s, 80s, 90s, which this data set was able to encompass. What we found is that the signature for brain aging seemed to be reversed in the individuals when they were on the ketogenic diet and exacerbated in those same individuals when they were on a standard American diet. Once we saw that effect, then we had to ask, okay, so what is it about the diet? So when you change your diet, you're changing lots of variables. You're changing what you eat, you're changing the macronutrients, you're changing perhaps also the caloric intake, even hydration, and so forth. And so what we wanted to do then was to isolate the variable of interest. So could it be that it was really just due to a very fundamental fuel difference between glucose and ketones? And so the second set of experiments tested people again under 3 conditions, but they came in fasting, and then we gave a scan, and on one day we would give them a glucose bolus, on the other day we would give them a ketone bolus. This was beta hydroxybutyrate type of ketone body. And what was important was there was a very clear before and after. So you don't have to ask what else happened during that week that they changed their diets, because really they're in the scanner, we test them, we give them a drink, we test them again. And it's very clear that whatever change there is before and after was due to the bolus. And because we were giving a bolus, we were able to calorically match it so that we knew that whatever change was occurring was not due to the total number of calories, for example, or other macronutrients, because the only variable that was changing was the type of fuel. We then wanted to ask, all right, well, is it really the subtraction of glucose or is it the addition of the ketones? So then the next iteration was to give people glucose and ketones versus glucose without ketones. And in this case, what happened was that the networks restabilized with the ketones, even if they'd previously been in a hyperglycemic state, which showed that it was actually the ketones, because the ketones seem to be able to override the deleterious effects of the glucose. That suggests actually something pretty interesting and optimistic, which is that individuals who are, for whatever reason, having difficulty following a ketogenic diet can achieve many of the same benefits by simply taking ketones. Now, there's a next installment in this, because then what we did was we have been following up with animal experiments to try to understand more precisely what's happening in terms of insulin resistance in a controlled manner. Can we take neurons? Can we make them insulin resistant? Can we give them glucose? Can we give them ketones? And what are the precise changes in terms of the firing dynamics, in terms of the communication along the axon between, synapses and so forth? And that whole series of experiments is being done by Nathan Smith at Children's National, who's one of our collaborators. And so what we're hoping is the next step is to be able to answer those mechanistic questions.
Very nice.
So there have been studies showing that ketones provide greater energy to cells than glucose, even when the fuels are calorically matched. And this benefit has previously been shown for the heart. So Lily, can you talk a little bit about how your study and experiments provided the first evidence for equivalent effects on the brain?
Yeah, this was actually something that, again, we didn't start off assuming at all. It was very surprising to me that healthy young individuals who were not in a deficit state, so we're talking about people in their early 20s who were not insulin resistant, the fact that their brain networks were stabilizing in response to the ketones speaks to, anecdotally, something that is often described by people who first go on a ketogenic diet, and they make remarks like, I feel like I think sharper. I think more clearly. And again, I think we have to sort of, to some extent, really discount those because there could be confirmation bias, there could be lots of things going on. But in this case, we could very clearly see that even in a non-deficit state, the networks were stabilizing. And that suggests that it is not that ketones are simply correcting for a lack of energy. So in a state of insulin resistance, what we expect and what we can see in the single neuron experiments is that the neurons lose the ability to have a usable fuel that then fuels the Krebs cycle and produces ATP. So the system in an insulin resistant state is going to produce less ATP, less energy for all of the processes that the neuron requires simply because it can't access the dominant fuel source, which is glucose. So if in fact the neurons were just simply insulin resistant and the ketones were able to bypass that insulin resistance and fuel the neurons, that would be already fantastic. But what was very interesting is that even in individuals, and we're seeing this also at the neuronal level, who are not insulin resistant, we're seeing more efficient communication simply after being administered ketones. And the explanation for that, we assume, although I would say this is still an open question, is work that Richard Veach had previously done with Karen Clark, showing that ketones produce more Gibbs free energy as compared to glucose, which means that even in a non-deficit state, the neurons presumably have more energy than they would otherwise. And then the question would be, well, what do they do with that excess energy? So I think the default assumption would be that they just excrete it, because if you have more than what you need, you just get rid of whatever the excess is. But it turns out that actually you do see a noticeable salutary effect, I would say.
The anecdotal reports of improved cognition that one often hears from people embarking on a ketogenic diet or ingesting ketone esters or ketone salts, that sort of parallels the experience that many cultures have and almost all religions have with fasting. When people fast, they almost always report improved clarity of thought. And of course, these people, if they really are fasting, they're in ketosis. And so it all makes sense even from that perspective.
And actually, in terms of the brain scans, we're seeing the same effects with fasting and administering ketones.
That makes sense to me.